First off, thanks everybody for joining. We're really excited to have Pranam kicking off, or Dr. Chatterjee is kicking off our first seminar of the semester. This is part of the Computational Health seminar series. It's intentionally virtual because we're trying to have people outside of Duke learn more about some of the exciting work we're doing here, as well as having people at Duke find out more about some of the work that's going on around the school. If you are local to Duke, keep in mind there is going to be a Meet the Speaker interactive coffee hour afterwards in Wilkinson in the first floor lobby. So definitely please come have some coffee, some pastries, and get to ask some more questions in person. But with that, I'll give a quick intro. We have a really exciting speaker today, so we're really lucky to have him talking to us. So Pranam Chatterjee is an assistant professor of biomedical engineering and computer science here at Duke. His lab is called the Programmable Biology Group. It's at the interface of computational and experimental design, specifically training generative language models to de novo design proteins that bind and edit target molecules. He graduated with all of his degrees from MIT, where he worked a lot on developing sequence-based algorithms to engineer broad targeting CRISPR enzymes. And his lab here is further establishing these deep learning-based methods that discover transcription factor proteins that can have direct differentiation of stem cells with a key focus on ovarian cell types. More recently, his research has extended into the emergent field of proteome editing, enabling targeting of proteins implicated in neurodegenerative diseases, viral diseases, and pediatric cancers. He's done a lot of work with translation. He's founded 2 different companies that build on this experience. He was recently awarded the Hartwell Individual Biomedical Research Award, as long as many other awards. So with that, I'm going to throw it over to him, and I'm really glad you've all joined us, and hope you enjoy the talk today.
Awesome. Thank you, Amanda. And thank you, everyone, for taking some time out of your day. As Amanda mentioned, my name is Pranam, and I'm an assistant professor of biomedical engineering and computer science here at Duke. And today I'm really excited to share with you the work that my team has done to develop programmable protein therapeutics using various algorithms. And that really comes to the first point of what our lab does. We integrate 2 very powerful platform technologies, one being algorithms and the second being proteins. And we develop very diverse algorithmic modalities, from bioinformatics algorithms, graph theory algorithms, and language models to discover, characterize, and engineer new and useful proteins. But not just any proteins. We like to develop proteins that we call programmable. Or, for example, proteins that can edit any DNA sequence, proteins that can bind and modify other proteins, as well as proteins that can make any new cell or tissue type. And we're going to go through all of these 3 things today, but we'll start here. Proteins that can make new cell and tissue types. The proteins I'm going to talk to you right now about are, I think, the most powerful proteins in our body, and those are transcription factors. These proteins are regulators of cell state, right? You know transcription factors. They can turn on and off the gene expression of other genes, but they can also regulate each other. They essentially define gene regulatory networks that can define a specific cell state. And so you can imagine that these are very, very important proteins, but they're also really good tools for cell engineering. You may know that you can take a somatic cell like a fibroblast, a skin cell. You can put in 4 transcription factors, and you can convert those fibroblast cells into stem cells, inducible pluripotent stem cells. That won the Nobel Prize. Now, a lot of work has gone into finding other transcription factors that you can put into iPSCs to differentiate them into cardiomyocytes, adipocytes, neurons, or pancreatic beta cells. But there is still one cell type that is really, really, really difficult to make. And those are germ cells, or in our case, oocytes. And you may be wondering what's so special about oocytes, other than the fact that they can underlie birth and having children, is that these cells actually undergo very significant developmental milestones. They are demethylated, they remethylate, they're meiotic, they're haploid. They're definitely not the easiest cell types to make. And so the question we've asked is, what are the sufficient set of transcription factors that can induce oogenesis from stem cells? But before we answer that question, we need to answer this one. How do we systematically identify transcription factors that can make new cell states from stem cells? To do this, we've created an integrated platform to help us discover these transcription factor proteins. As you'll see, we always start with an algorithm. And in this case, we utilize a graph theory algorithm that can say, hey, here's data, here's RNA sequencing data of every step in a differentiation process, whether it's oogenesis, cardiomyocyte differentiation, or other cell types, and convert that data into a graph via a gene regulatory network inference algorithm. Not something that we had to discover. But what we did was we were like, well, we have this graph. What are the important nodes, points in the graph? And say, hey, those nodes are likely important genes or transcription factors that drive and essentially define this graph. And we found that once we were able to identify these central transcription factors, we saw that our algorithm was very accurate at finding the critical transcription factors to make neural stem cells, myoblasts, melanocytes, and other somatic cell types. But you may not know these before the fact. So to actually take these transcription factors and put them in cells, what we did was combinatorially screen them. Meaning, hey, that cell number 1 got these 5 factors, cell number 2 got these 3 transcription factors, and such that we can combine different combinations of our prioritized proteins into cells. And then by a single cell sequencing, we can be like, all right, that cell got these 3 transcription factors. This is what that cell looks like. This is its molecular phenotype. And so now with this integrated method, you can identify transcription factors that drive a specific phenotype or cell state of interest. So we call this method stamp screen. And if you want to read more about stamp screen, we published it a few years ago as a general tool to help you discover transcription factors to make whatever cell type you want. We even developed a cloning method where you can be like, all right, I want to try these 5 transcription factors. I can easily build them, put them in a cell, and see what happens. For us, we use StampScreen to identify the transcription factors central to oogenesis. We took a data set of natural oogenic differentiation, which is a long process. We defined a gene regulatory network of that process, and we used our algorithm to identify the core transcription factors that were central, right? PageRank is a centrality algorithm central to the differentiation of oocytes. We found that those transcription factors had very critical roles in germ cell development. They were important for developmental processes, reproduction, oogenesis, cell differentiation. But really, we needed to see if these actually worked. And so we had to see first, are these transcription factors well expressed in fetal gonads where these oocytes arise from? But more importantly, can these transcription factors drive oocyte formations in vitro? So what we did was develop a germ cell induction protocol, right, to screen these transcription factors. And to do this, we basically said, hey, here are growth conditions that can promote germ cell formation. Now, if we put in our transcription factors, do these cells go to an oogenic-like phenotype? And to track that, we had reporters where if you turn on this gene, DDX4, that is only turned on when the cell has achieved an oogonia-like state. Nanos3 is another gene that is only turned on when you achieve a primordial germ cell or a more intermediate state. So we could track, hey, are we creating the cell type we wanted? What we found was 3 transcription factors from our algorithm that drove the expression of DDX4, which is this oogenic like gene. And so these 3 transcription factors, when we put them together and then put them all into every cell, we found that we drove much higher yields of DDX4 expression, meaning we were making more cells that had this oogonia-like phenotype. How do we know these are oogonia-like cells? Well, we first examined the gene expression profile of those cells, and we saw that not only DDX4, but all the genes that we would expect to be in an oogonia-like cell, we were able to see high levels of gene expression in those genes in our cells. So we know that we're making, at least on a transcriptomic level, similar cells to oogonia. We also confirmed this at the methylation level. This is a PCA of our methylation profiles. And you can see that our methylation profile showed that our cells clustered very similarly to fetal germ cells, which is the methylation profile that you would like to see. Also, we showed that once we take those cells And we're able to then put them back in culture, that these oogonia-like cells, the ones that express DDX4, not only did they grow and expand, they also continued to express that DDX4 gene, meaning that these are sustaining an oogonia-like profile. So we also use STAM screen, right? That's really a really cool story. We hope that'll be communicated soon. But we've also used STAM screen to identify 3 transcription factors, DLX5, HHEX, and FIGLA, that drive improved germ cell formation. And so these are the intermediate germ cell phenotypes. They're really hard to make in cells, but we showed that by just adding 3 transcription factors, we can drive increased levels of production of germ cells, which allows now biologists to study reproduction much more strongly. By the way, we've used STAM screen to also generate all the ovarian cell types as well. We've been able to create a human ovarian organoid from stem cells just by transcription factor overexpression. And that work has been published. And this has all led to a company that Amanda mentioned, Gamito, which we founded, which essentially utilizes iPSCs, utilizes our cell engineering methods I just showed to you to create the organoid of the female reproductive system such that we can use this platform to generate fertility solutions for female patients. And what specifically Gamito does, and I'll just give it a very brief background of our work, we basically use iPSCs, we differentiate them into these ovarian support cells like granulosa cells, theca cells, stromal cells, and we incubate immature oocytes from patients, and we're able to then mature those into fertilizable mature M2 oocytes. And this work is currently in phase 3 trials. And we've shown that in the company, we've been able to get oocytes that demonstrate improved maturation capabilities than standard methods, and also display very similar transcriptomic and epigenomic similarity to real M2 fertilizable oocytes. So that's really exciting work, and hoping to see this in the clinic very soon. And so I started this company alongside my 2 co-founders a few years ago, and now it's become a really nice and really one of the bigger femtech companies currently in the world. Great, so that's our story, on a very short story on our cell and tissue engineering applications using proteins, but I want to come back in the central dogma back to the core molecule, and that's the ability to edit DNA. This is the idea of utilizing a tool like CRISPR-Cas9, something that we're all very familiar with, to target and cut and edit DNA sequences. And CRISPR is an amazing tool. As you can see, you really only need 2 parts, a guide RNA to target a sequence and a Cas9 that can be guided to that sequence to be able to edit. And that won the Nobel Prize, obviously, but there's a problem. CRISPR-Cas these enzymes need 2 Gs, right? A, C, G, and T for DNA. You need 2 of Gs beside the target sequence. We call this the PAM, to be able to bind and to edit. And you may be like, well, that's not a big deal, Pranam, right? You'll find 2 Gs somewhere. But that limits you to only about 12.5% of all DNA sequences. And so how do we fix this problem? So to do this, we said, hey, let's find new CRISPR proteins in nature that may have relaxed or less restrictive PAM sequences so we can edit more DNA sequences. So we developed an algorithm to find these CRISPR proteins with less restrictive PAMs. We did this by searching for PAMs by aligning bacterial genome sequences to that of phage genomes. So just a purely sequence-based approach. We called it the search for PAMs by alignment of DNA sequence targets, or we call this Spamalot. And Spamalot we've utilized to discover a host of new CRISPR proteins that may have broader targeting capabilities. Here's a few examples. So Spamalot helped us to discover first an enzyme called ScCas9. ScCas9, unlike the original SpCas9, only needed 1 G. And when you need 1 G, you can edit up to 50% of all DNA sequences. We even optimized ScCas9 with 2 insertions from other bacterial Cas9s, discovering a Cas9 we call SC++, where with high activity and very low off-targeting, making it a very good genome editing tool. Finally, we were just using Spamalot. we were the first group to find a Cas9 that didn't need in either a G or a C in its PAM, meaning that now we can edit a whole new host of DNA targets by having a Cas9 that only needs A's or T's in its PAM sequence. And we call this enzyme iSpyMac. And look, don't take my word for it. Take the word of all the people around the world that have utilized our Cas9s to edit the genomes of rabbit, of rice, of wheat, of mutant human embryos, and even the current trials for spinal muscular atrophy correction using CRISPR are utilizing iSpyMac as the base enzyme. So we're really excited to see all of this work continually translationally. But while we were doing all of that work, my very close friend and collaborator, Ben Kleinsteiber, had been engineering the original Cas9 using directed evolution to get a broader PAM targeting capability. He found an enzyme called SPRY, which only needs an A or a G at position 2 to be able to bind and to edit. And that's the broadest Cas9 we knew, R being A or G. He called this enzyme SPRY, and this really inspired us to think, can we actually get to a Cas9 that doesn't have any specific PAM requirement, meaning we could go to all DNA sequences. So to do this, we took SPRY and we took SC++, our very optimal Cas9. And as you can see, remember, the PAM of SC++ is NNG. The original one was NGG. So we removed the dependence on the second base. SPRY's PAM dependence is at position number 2. So we decided to take the best parts of SPRY and the best parts of SC++ and mash them together to create an enzyme we call SPRYC. And we've shown that SPRYC, utilizing the most optimal components of SPRY and the best components of SE++, which indicates this specific loop-like structure, we've shown that SPRYC has almost full PAM flexibility, meaning no matter what combinations of bases you have in the PAM, we're able to go and edit there. And Lynn and Sabrina have shown in the lab that we have the SPRYC has fewer off-target effects even than SPRY itself, even though it is broader, and that SPRYC now, because it is so broad, can access and edit and fix mutations in various diseases that were previously untreatable, such as Rett syndrome, this C to T mutation that we can make, as well as the hard-to-target repeat region of the Huntington protein, the Huntington gene. We did not have ability to edit it. Now with SPRYC, we're able to make therapeutically relevant edits to this sequence. So it's a fully PAM flexible Cas9. You guys should use it. It's on AdGene. It's here. It was published about last year. And now what we're doing is applying SPRYC to many previously untreatable genetic disorders like Klinefelter's or microdeletions or cystic fibrosis. We're hoping that SPRYC can be utilized to fix these genetic abnormalities and hopefully eventually cure these diseases. So that's our work that's ongoing and has been completed to edit DNA in a very programmable and sequence unrestricted fashion. So the rest of the talk, though, will be something I'm really, really excited about, something that our lab is primarily focused on, and that is taking this programmability of CRISPR, right? The ability to edit almost any DNA sequence and apply it to proteins, right? Can we edit proteins, the actual things that cause disease? So what our lab does is we computationally design peptides, small proteins that can bind to other proteins inside of the cell. And what we do is we don't just bind, we actually say, all right, here's an enzyme called an E3 ubiquitin ligase. It naturally binds something. We take out that binding domain and we put in our peptide. So we essentially attach our peptide to the catalytic domain of enzymes like these E3 ligases, such as CHIP. And what that does is that allows us to not only bind, but add this disposal tag, this ubiquitin tag to proteins, enabling the targeted protein degradation in the proteasome. So now we could potentially not only bind, but degrade bad proteins inside of the cell. We call these genetically encodable peptide E3 ubiquitin ligase fusions. We call these ubiquibodies. And we designed our first ubiquibody to degrade SARS-CoV-2 viral proteins. The story goes that I was working on CRISPR, I was in the lab, this is right when I was about to start my lab at Duke, and we were like, hey, instead of targeting the RNA or the DNA, what if we just got rid of the viral proteins? And so we developed ubiquibodies to degrade viral proteins. And what happened was that when you introduced these ubiquibodies into viral cultures, the infectivity of the virus reduced by over 60%, telling us that not only did we degrade the proteins, we actually had a functional effect on the virus itself. So, but this really got us excited. We could easily degrade these viral proteins. Can we design a ubiquibody to any target protein, any bad protein, and a protein that is dysregulated? This would be an incredible tool because our goal, like I just said, is to create a CRISPR-like system to edit proteins. You guys know CRISPR by now, right? You can take a target DNA sequence you want to edit, make a guide RNA that binds to that sequence. You can have a Cas9 that complexes with the guide RNA and is able to go to that sequence and edit it, right? Pretty simple. But what if you could do the same for proteins? What if you could give me a target protein sequence? You could design a peptide that binds to the protein, attach it to an E3 ubiquitin ligase, and now you can bind and degrade that protein, right? That would be the analogous system to CRISPR. But how do we achieve this level of modularity? Because look, for DNA, it's easy, right? You just have to match the guide RNA to the target DNA, and it's just complementary base pairing, and you got it. But for proteins, it's not so simple, right? A lot of proteins that cause a disease, one, don't have binding pockets, meaning you cannot design a small molecule drug to this protein. No matter how hard you try, there's no spot on the protein that that small molecule will dock to, all right? And worse, these proteins are very disordered, meaning if you wanted to look at the structure of a protein like PAX3-FOXO1, which drives pediatric cancer, this is not going to tell you anything. Look at that protein. It looks like spaghetti, right? So you cannot use small molecules or structure-based methods to hit these very bad but important proteins. And so we need a new strategy. And the question we've asked is, can we design guide peptides, right? Not guide RNAs now, guide peptides that can bind and degrade undruggable target proteins without needing their structure. I know that's a difficult task. And you may be like, Pranam, that sounds almost impossible. How are you going to do it? Well, we turned to something that you may not expect. And those were natural language models. You guys know these models, right? Some of you use it for your problem sets, ChatGPT, Claude, Llama 3, if you're using the open source models. The way these models work is that they are trained on millions and millions of text examples. You're not telling them what type of text it is or the semantics. No, you're just giving it text. And by training on all this text, these models have billions of variables that are learned. And those variables encode syntax, semantics, grammar, and a bunch of other properties of that text, such that if you want to generate a new text, you have that information present. What we do is we train protein language models. Same thing, we train on millions and millions of protein sequences. And these models like ESM or ProGPT or ProT5, they're not learning syntax, semantics, and grammar. They're learning structure, function, interaction, and a billion other properties of these proteins. Our favorite protein language model is this model called ESM. And this was trained by Facebook, or now Meta, that said basically it's actually quite simple. The idea was, here's a sequence that goes through the model. To train the model, we hide portions of the sequence. The model's goal is to remake that sequence. That's it. This is an idea called masked language modeling. And by doing that, over 250 million protein sequences, and to do that well, the model has to learn properties governing structure, function, interaction. So you get a representation of the protein that has learned its critical inherent properties. And that information, the way I know this, right, is you can take that information and you can say, all right, I'm going to keep that information present. Now let me use that information to predict structure. And you guys may know AlphaFold, but ESMFold is a very good protein structure prediction algorithm that uses these language model embeddings to get to a final 3D structure. So it tells you that these language models have a lot of information, but not just language models. I'm sure you've also heard of image generators like DALL-E, right? You can give it a text of a corgi playing a flame-throwing trumpet, and you can get an accurate picture of a corgi playing a flame-throwing trumpet. And the way this works is that we say, hey, we have good representations of text via language models, and we have good representations of images via an image model. Now, let's join them in a shared space such that a text of a cat and a picture of a cat are close together, and a text of a cat and a picture of a dog are far apart. So it learns those spatial interactions. We call this contrastive language image pre-training, or CLIP. And so instead of text and images, we trained a CLIP model for peptides and proteins. And to do that, we basically utilize ESM encoders to represent these sequences to the model. And the model was like, all right, this peptide binds to this target. That should be a high score. This peptide doesn't bind to these targets. That should be a low score, and it inherently learns those properties. But you got a new target, and you don't have a binder to that target. Where do you get these peptides, right? So really the question is, we need to generate de novo peptide candidates that are specific to our target protein of interest. To do this, we were like, well, let's use these language models. The idea is we have real peptides, right? They exist. They don't bind to our target, but they are real and they have good properties. Let's put them into this language model space. Let's perturb their representations, right? This is all mathematical. And then let's use those perturbed representations to get new protein sequences or peptide sequences. Now we have a bunch of new peptides, millions of new peptides, but they maintain the naturalistic properties as defined by this pre-trained protein model. So now, all right, cool. I have a lot of new protein peptide sequences. I can throw them through CLIP. And now we have this new model called peptide prioritization with CLIP, or PepperClip. And what we've shown is that PepperClip can prioritize target-specific peptides. Take a look here. You have peptides and proteins that are known to bind. These are not in our training set. And you can see that their scores for CLIP are really high. They should be high because they bind. But then we mismatched peptides and proteins that don't bind. They're just randomly mixed and matched together. And you can see that their CLIP score is like 0, right? So we know that our model is accurate at defining what is a real peptide protein interaction. And so but now the real proof is in the pudding. And so to do this, we have this interesting philosophy in the lab. We're like, all right, we can test these in our lab, and you'll see a lot of experiments where we do. But more importantly, if I gave it to someone else who's working on a new protein, and they used our model versus the best structure model, remember we're using sequence, if we give sets of both, which ones do better, right? So we started with this protein called Ultra ID, which is an enzyme, very structured enzyme. And as you can see in blue, you can see reduction of the activity of this enzyme, meaning we bound to it and we inhibited it. And our best inhibitors were not from the structure-based RF diffusion model, but rather from our sequence-based model. No structure needed at all. We also showed, well, what if we moved to proteins that are more functionally relevant in cells? Beta-catenin mediates the most important transcriptional signaling process in cancer. What if we could target that? And we showed one that when we attached our peptides to the E3 ligases to create these ubiquibodies, we were able to degrade endogenous beta-catenin, meaning we depleted beta-catenin in cells, as you can see here. Beta-catenin went away. But not only that, we also showed here on the left that our beta-catenin targeting ubiquibodies from PepperClip was able to reduce the cancer signaling in these cancer cells, meaning we're not only degrading the protein, but we're having a functional effect. And this is because we bound to beta-catenin tightly. The affinity of our ubiquibodies to beta-catenin were about 100 to 200 nanomolar, which is really, really tight for a peptide. But then you may be asking, what about these proteins? What about proteins that are completely disorder, right? There's no structure available. I can't even run RF diffusion or AlphaFold or anything on these proteins because they're so disordered. Can we bind to these? This is a protein that causes a devastating childhood cancer called synovial sarcoma. And what we showed that when we tested 10 with our collaborator at San Diego, Ani Deshpande, he found one out of 10. He found one that was able to hit this protein and degrade it by over 50%. And when he put these degraders into cancer cells that natively express SS18, SSX1, we were able to see knockdown completely of this fusion protein. Again, this is a protein that has literally never been touched by any pharmacologic agent. And that shows you that language models are so good. So hopefully I've shown you that language models are good, unconditional generators of peptides. Remember, we didn't give it a target sequence to make the peptides. We just made them, and then we saw if they bound to our target sequence. But what if we could flip that question on its head? What if we could generate peptides conditioned on the target sequence? This is somewhat similar to ChatGPT, where you give it a question, and from that question, it makes an answer. It's not like you made a bunch of answers and you screened it through ChatGPT to find the one that was correct. That would be similar to Paperclip. So we created, or Leo in the lab, our master student, brilliant guy, created a model called PepMLM, or peptide generation via masked language modeling. It's actually not that complicated. I'll tell you how it works. So the way it starts is we have a data set of a bunch of peptide protein interactions, right? This is a peptide, this is a protein, this is a protein, this is a peptide. We have all these different pairs. What we do is we position the peptide sequence at the C terminus, or the right end of the target protein sequence. And then we hide it completely. Not part of it, we hide the entire sequence. And the model is like, what the heck, right? So we give it to ESM, an existing protein language model, and we're like, ESM, try to remake this sequence. And the model at first obviously has no freaking clue what it needs to do. But it learns, right? It learns to do this better. And we do that by updating the weights, the parameters of the model, such that eventually it gets really, really good at saying, all right, here's a target protein, here's a hidden binder, make the right binder. And it ends up doing so, such that now you have a new protein, right? You have a new protein sequence, not ever seen in this training set. We can put that target protein sequence into PepMLM. We say, all right, you want a binder of 5 amino acids or 20 amino acids, mask, 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 mask. And now the model is able to say, all right, here's your 20 amino acid binding sequence, right? Because it learned to do that very well. The big question then is, what is the hit rate? Meaning it's going to make peptides. Do they work? And what percentage will they work for you? So to do this, we actually wanted to make it hard on the model. We were like, we calculated the hit rate by saying this. What percentage of target proteins, we looked at diverse targets, is PepMLM's generated peptide better than a known peptide that has already been engineered to bind to that target, right? It's a fair comparison. If we're better than something that has been crystallized or SPR'd to that target, and we are better than that, we know we made a good peptide. PepMLM's hit rate was over 38%, meaning 38% of targets, we made a better peptide than one that was engineered for years to that target protein. And remember, these proteins are structured. So we tried RF diffusion, the competitor model. It gets the structure of the protein and its hit rate is 30%, while our model only needed the sequence, and we were able to get a higher hit rate on these structured target proteins. But you may be saying, well, what about experimentally, does this hold up on more difficult targets? We showed one that PepMLM peptides bind more potently to proteins like receptor proteins like NCAM1, better than RF diffusion. And we said, all right, cool, but let's go to some hard targets, like disease-causing targets. We looked at 2 proteins that drive Huntington's disease, right? We talked about it previously, a very devastating genetic disorder that creates these bad Huntington proteins. And there's a protein that can regulate Huntington protein production called MSH3. And we showed that our PepMLM-derived ubiquibody degraders potently degrade MSH3 and even mutant Huntington itself, which is the core protein that drives this disease. We can see, we just tested 5, and we were able to find multiple that depleted Huntington protein in Huntington cell lines. By the way, we've also shown that PEP-MLM-derived ubiquibodies can degrade emergent pandemic viral phosphoproteins. You think COVID was bad? Wait till Nipah virus or Hendra virus comes out. And so we are like, well, we're just going to go ahead and bind and degrade those viral proteins, and we've been able to do so very effectively, over 50% these viral proteins at one point. By the way, those phosphoproteins are the proteins that mediate the infectivity of these viruses. We've even done it for HMPV as well. As you can see, we have degraders that are close to 80% degradation capability. So hopefully I've shown you that with these tools, by combining generative language models and protein engineering with these enzymes, we have a programmable system for programmable targeted protein degradation. And we have a company called Ubiquitics that I've started that is pursuing more challenging cancer targets to translate this technology. So wait, though, all of this work leverages ubiquitination to degrade proteins. That's great. If your protein is bad, you want to get rid of it, right? But what if you have a protein that is improperly ubiquitinated and that protein is being removed, but you don't want it to be, right? How do we fix that? Here's an example, P53, right? It's a tumor suppressor gene. You need that protein there. If you degrade that by ubiquitinating it, which happens in breast cancer, guess what's going to happen? P53 is going to go away and you're going to get cancer. So could we stop? Could we remove this ubiquitin tag from P53, right? This would be the holy grail question. So instead of ubiquitinating target proteins, can we use deubiquitinases to remove and stabilize useful target proteins with deubiquibodies? What deubiquibodies do and what we've made is we link a potent deubiquitinase domain, just like we did for E3s, to these PEP target binding domains. And we've shown, Lauren in the lab, or my PhD student, she showed the most optimal deubiquitinase was this protein called OTUB1, which we found from this beautiful paper that screened a bunch of deubiquitinase and E3 ubiquitin ligase domains for activity. So we found one that was very good when we attached it to binders. But what if we can now attach these to our language model derived peptides? We first showed that when we took our beta-catenin targeting peptide, we did this from our previous algorithm called salt and pepper, and attached it to OTUB1. You see, right? OTUB1's level, beta-catenin's level went up, right? Bars are going up now, not going down, because we're now stabilizing. We were able to get that. Pepper clip peptides, we put them in deubiquibodies, and we can stabilize a tumor suppressor kinase called WEE1. You can see very nice stabilization of WEE1. And guess what? I think the coolest of them all, we were able to make peptides to P53 using PEP-MLM. We attached them to our deubiquitinase domains, and we were able to stabilize P53 in cancer cells,
There's a few
telling
boxes
us
that
that these
are still
cells...
here.
Oh,
But
someone's online. And we were able to then stabilize P53 in these cells. So that tells you that the system is super modular to make binders and put them into our deubiquibody architecture. So now we can programmably degrade proteins, we can stabilize them. The next steps that Lauren and the team are working on are to phosphorylate proteins or to dephosphorylate or acetylate or glycosylate. If we can take these peptide fusions, put them into a delivery vehicle, and inject them into animals or eventually humans, we have a very, I think, precise and specific therapeutic platform. We're working on that in the lab. I cannot show you this data in this talk, but we have mouse data showing that we can degrade important cancer-causing proteins in mice and get a therapeutic effect. So we've built a programmable RNA-guided genome editing with our CRISPR enzymes, but we're also building a programmable peptide-guided proteome editing platform with our ubiquibodies, our deubiquibodies, and our new technologies. But there is another reason why you don't want to do what I'm showing you with small molecules or your standard drug modalities. I really don't think small molecules are good drugs, and I'll tell you why I think that, right? One, they're not modular or tunable. It's hard to make a small molecule. It takes years to define it, to chemically synthesize, to screen. All of that can take a long time. but they're also not selective, meaning I want to bind to a specific spot on a protein, or I want to bind to a specific version of the protein. I'm not able to do that because I still have to bind to that binding pocket, which may not even exist, right? What we do is we have that capability in theory, right? We should be able to bind to a specific spot on a protein, not just anywhere, but somewhere specific. And we should be able to tell the difference between a bad version of the protein and a good version of the protein, even if that difference is really, really small. So first, let's just make sure we can go to specific spots on a protein. Because look, all of our current peptides from Pepperclip, from PepMLM, they bind well to proteins, but we can't tell them where. We just say, here's a sequence, make a binder. I don't know if it binds here, 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 right? So let's fix that. Because this may be important, right? For p53, we may want to bind to the DNA binding domain, which mediates its transcriptional activity. We may, for the amyloid precursor protein that underlies Alzheimer's, right? We may want to bind to this beta secretase cleavage site, which allows for the amyloid plaques to eventually form. And finally, for fusion oncoproteins, we may not want to bind anywhere on the protein. We may just want to bind to the breakpoint that defines this fusion, right? So binding to a specific spot on a protein matters. And so the problem is there are methods to do this using structure. RF diffusion is a great tool we've shown you. You can actually specify where on the protein you want to bind. But what if you need to bind to these disordered proteins? You cannot say bind here and it will bind because it's disordered. The algorithm has no idea what it's looking at. So what we've done to fix this is we've developed MOPIT, a motif-specific PPI targeting algorithm. To do this, we first need to predict which amino acids on the target protein does a peptide or a protein bind. If you give it a protein, you give it a target, where does it bind, right? We have many examples of training data to do this. Over 300,000 validated protein-protein interactions, and we know where they bind. And so we developed an algorithm called Bind Evaluator that can predict the binding residues provided the target and the binder protein sequences. As you can expect, Bind Evaluator uses these protein language model embeddings. It says, all right, let's take these representations, let's process them through a CNN. So that in this case, the convolutional neural network is learning local interaction information on the target sequence. So we understand how that binding should occur. We then combine the binder information, the target information via this reciprocal multi-headed self-attention module, basically to understand the dependencies between the binder and the target, and we're able to predict the binding sites. So Bind Evaluator performs very strongly on learning the binding sites. First, within protein-protein interactions, which we have 300,000 data points for, you can see we have very good accuracies. But when we do it with peptide proteins, which we only have 12,000 data points for, we transfer learn to peptide proteins, and we get a very, very good AUC and accuracy at predicting. MOPIT then says, all right, I have PepMLM, right? So I can make starting peptides. Now I have Bind Evaluator, which can tell me where they bind. Let me optimize the sequences to now bind to that specific spot, validated by Bind Evaluator. So we use a genetic algorithm to update the sequence based on Bind Evaluator predictions to create new peptide sequences from original PepMLM targets sequences. MOPIT peptides bind very well. We did the same analysis. Do we bind better than known peptides to targets? We did really well there. And we also wanted to see where our generated binders go. And so we use this tool called PeptiDerive, which evaluates the interface energy score of amino acids on the target protein. And we showed that when we bind to a specific spot, right, we have a higher relative interface score. So let's see where our peptides bind. We showed for first on just nice structured targets. If you look at the heat map, we wanted to bind to the things in red, and the dark blue is where it actually bound, right? So we were able to bind specifically to those regions. Here's disordered domains on structured proteins. We were able to bind, we wanted to bind to these 6 amino acids. Look where we bound, great. We say, what about semi-disordered target proteins? We wanted to bind to these amino acids. We were able to bind there very effectively. And also other disordered proteins like MIND1, we did that very well. And even completely disordered proteins like EWS-FLI, we were like, well, we wanted to bind to these amino acids. That's where it bound. So we are really confident that MOPIT is going to work in the lab. We're still working on that, early days, but we're really excited about this progress and we wanted to share it with you. So now you can take a target protein sequence, say, here are the amino acids that I want to bind to, and MOPIT can make that binder. Great. So we can bind to specific spots on target proteins. I want to show you 2 quick examples of how we can be what we call isoform specific. Because look, proteins can come in multiple different versions. Like, what are these things, right? These are phosphate groups. These are called post-translational modifications, which alter protein structure and function. They also enhance proteomic diversity. So now you're not just dealing with the subset of wild type proteins. I can modify them and make them do completely different stuff. Here's an example, right? Take STAT3. STAT3 is a protein that if it's phosphorylated, it causes cancer. But once it's not phosphorylated, it's a good protein. You need STAT3 present. So we want to degrade phospho-STAT3, but not the wild type STAT3, which is an important protein in your body. Right? So can we bind specifically to PTM'd disease-driving targets, but not their healthy wild-type counterparts? But there's a problem. Current models only encode wild-type amino acids. They don't even know what a phosphoserine is. They'll just call it a serine, right? So what we should do is incorporate post-translational modifications into these models so that we can then design with them, right? We did this. We call this PTM Mamba. And what PTM Mamba does is it utilizes this very new AI architecture called Mamba, which can model sequences much more efficiently in a computational setting, right? So it can take the same attention mechanisms of transformers, of language models, and just make this process more efficient. And then what we did is we turned it bidirectional, so that now we learn information from left to right and right to left and all the dependencies in between. And we fuse the information from that Mamba block with ESM. So we don't lose the natural protein information. We just add the post-translational modification information. And what we've shown is those new embeddings that incorporate these post-translational modifications learn the properties of natural sequences, but when you add that PTM to the sequence, the embedding shifts. Previously, if you said, here's a protein and this one amino acid is phosphorylated, it'll be the same thing. There would be no difference. Now we can tell the difference, and we show that our model focuses on these post-translational modifications. You can see that the phosphotyrosines and phosphothreonines are close together. The acetyl modifications are close together. The model understood those differences. We show that PTM Mamba can outperform models on phosphorylation site prediction, on acetylation site prediction, on disease association prediction, as well as druggability. And the point I'm saying is that now that the fact that we can incorporate post-translational information into our models, we have more ability to distinguish functional features. And so now you can give it a new sequence and you can even say, hey, I want to know what PTMs are on my protein, and the model will be able to tell you that. Great. And our goal is to take PTM Mamba and now design binders to proteins that are post-translationally modified, which they are the ones that drive disease. Great. And be specific. The last thing I will say very quickly, so we'll still leave some time for questions, is what about these weird proteins I've been showing you, right? These proteins have 2 proteins together. These are called fusion oncoproteins. These are unnatural cancer-driving proteins that contain domains from 2 natural proteins, right? 2 very normal proteins. But when you put them together, they're very disordered. Look at that. Again, looks like spaghetti. Like PAX3 FOXO1 is made up of PAX3, which is a normal transcription factor, FOXO1, which is a normal transcription factor. I told you how important transcription factors are. When you add them together and they mash together in cancer, they cause this cancer-driving mutant protein, right? So you don't want to touch PAX3 or FOXO1. You only want to degrade PAX3 FOXO1. You only want to target the fusion protein. But look, our models like salt and pepper, paperclip, PepMLM, they've never seen fusion oncoproteins. So they don't even understand what these proteins are. And so Sophie in the lab decided to change that, right? She said, because ESM has never seen a fusion, it may not generate optimal representations for fusion oncoproteins, and that may limit our ability to design binders to get rid of them. So she created Fusion PLM. It's the Fusion Oncoprotein Aware Protein Language Model. Basically, all we did was we said, here's ESM. Let's put in fusion proteins into the model and update it. But instead of just randomly doing mass language modeling, she focused her attention of the model on the regions that were critical for the fusion oncoproteins to interact with their cancer-causing counterparts. And so we were like, maybe the model can focus on these amino acids. And we showed that when the model does that focusing, we get basically after training on with over 40,000 of these sequences, the model outperforms all other models at understanding fusion oncoprotein properties like puncta presence or localization of these proteins. We also show that they understand the actual physical chemical properties of fusion oncoproteins very well. You can see the R squared values are super high with our models, right? These fusion PLM models. And that we can even predict which amino acids are disordered on a protein just because we trained with these disordered target proteins. So take a read of Fusion PLM. We're going to now take our PepMLM model, replace ESM2 with Fusion PLM, and now we should be able to make binders to get rid of these pediatric cancer-driving proteins. And while we're on the topic of specific targeting, very quickly, we've developed new models in our lab that can enable binder design not just to proteins or DNA. Well, first to DNA. We've made DNA binder protein generating models with just language alone, as well as to heavy metals. Now we can bind to like copper, cadmium, lead, so that we can sequester them in nature and not have them pollute our environment. And we're now even having promising results to bind the most unbindable thing in nature, PFAS, which I think is going to cause a lot of diseases in the future because it's in all of our bodies and it's undegradable. So we're going to make a binder and a degrader to these molecules. I know that's a lot, right? But I hope I've shown you that with generative sequence-based binder design, right? That's what we do. Our lab can program nature to do what we want it to do, to get rid of an undruggable protein, to mutate a DNA sequence, to target a heavy metal, or maybe even to bind and degrade pollutant chemicals. So with that, I want to thank my wonderful lab who does all this work, not me. I just support them. I'm their cheerleader and funder. But the idea is that we have a great computational team that does all the AI and an experimental team that not only validates the model, but also builds really cool tools with the binders that they design over here. We have great funders that have believed in us and, from the early point and said, hey, we can trust this lab to make all this work happen. And also to our kind and supportive collaborators who say, all right, we're going to test your stuff. We don't know if it'll work, but they end up seeing it does. And they've really worked closely with us to show the utility of our proteins. And thank you to you guys for listening to our story. I know we're a young lab. Not many people know us. Maybe if you read Twitter, you'll probably know me very well, unfortunately. A little too controversial there. But I hope you'll continue to follow our work. And so if you really want to know more, come to our website. We're going to continue doing this cool stuff. And yeah, I'm happy to take any questions if you have them. Thank you so much.